Welcome. This video will explain how the Renin Angiotensin Aldosterone system, or RAAS, functions to maintain homeostasis. RAAS is an integral part of the urinary system and a homeostatic mechanism which aids in the regulation of blood pressure and sodium plasma concentration. The system is activated via a negative feedback loop triggered by three main factors. A drop in blood pressure detected by baroreceptors in the carotid sinus, decreased renal perfusion detected by baroreceptors in the afferent arteriole of the kidney, or a decrease in sodium chloride concentration in the distal convoluted tubule detected by the macula densa within the nephron. A signal is then sent to the juxtaglomerular cells located proximal to the macula densa. These cells are also sometimes referred to as granular cells. When the signal is received, this causes the release of renin from the juxtaglomerular cells, also known as granular cells within the kidneys, into the bloodstream. Renin is both a protein and an enzyme and serves only one purpose. It functions by reacting with or cleaving a protein called angiotensinogen when they meet in the bloodstream, ultimately creating the hormone angiotensin 1. Angiotensinogen is a precursor to angiotensin 1 that is constantly produced by the liver and secreted into the bloodstream. The newly created angiotensin 1 is then met within the bloodstream by angiotensin converting enzyme, more commonly known as ACE, which is produced primarily in the lungs, and therefore, like angiotensinogen, is not secreted based upon a trigger. ACE functions by making a slight modification to angiotensin 1, converting it to angiotensin 2, which is where it gets its name from. Until a research study was conducted by some researchers in Melbourne in 1992, it was thought that ACE was synthesized solely in lung capillaries. This research has shown it is actually produced throughout the entire body in much smaller quantities, such as in blood vessels and all endothelium. Unlike angiotensin 1, which serves no biological function except obviously as a precursor to angiotensin 2, angiotensin 2 has four main effects: vasoconstriction, aldosterone secretion, antidiuretic hormone release, and sympathetic nervous system activation. We'll now take a look at each of these in more detail. Angiotensin 2 is a natural vasoconstrictor. It signals the tunica media or smooth muscle layer of blood vessels to constrict, therefore increasing blood pressure. It should be noted that vasoconstriction is most effective on arterioles rather than venules, as the tunica media of veins is much smaller or thinner than that of arteries. This is the most fast-acting effect of angiotensin 2 and happens very rapidly as soon as angiotensin 2 comes into contact with the smooth muscle cells of the tunica media within blood vessels. The second function of angiotensin 2 is sympathetic nervous system activation, primarily creating a thirst response, and plays a further role in aldosterone secretion, which we'll learn more about soon, and vasoconstriction, among other sympathetic nervous system. Is stimulation of the adrenal glands pictured here, which are located superior to the kidneys to produce aldosterone, a steroid hormone. The function of aldosterone is to stimulate the increased secretion of potassium and hydrogen ions through urine, increase water reabsorption, and the levels of sodium in the bloodstream. Therefore, blood volume is increased, and consequently so is blood pressure, aiding in the stabilization of the internal environment. Angiotensin 2's fourth and final function is to trigger the release of antidiuretic hormone, known as ADH. ADH also goes by the name vasopressin. ADH is produced in the hypothalamus, a gland that forms part of the diencephalon within the brain, and is stored in vesicles within the nearby posterior pituitary gland until released. ADH is not synthesized based upon a RAAS trigger and is produced only when stored levels are low. When triggered for release by angiotensin II, it is secreted from this gland into the bloodstream by the posterior pituitary and functions in three ways. Increased sodium absorption within the ascending loop of Henle, which is part of the smallest functional unit of the kidney, known as nephrons. Increased absorption of water from the distal convoluted tubule within the nephron, and finally, opening of aquaporins along the collecting duct of the nephron, allowing for much greater water reabsorption. The consumption of alcohol disrupts the stable environment maintained by RAAS. Alcohol itself inhibits the secretion of ADH, which we have just discussed aids in the function of fluid retention. Therefore, when this is inhibited, the kidneys do not retain or reabsorb fluid, instead producing more urine. Further, it is worthy to note that this will cause dehydration. Here's an overview of the entire renin-angiotensin aldosterone system.